So today we're continuing with Unit 1, and we'll be on page 8 of your notes. Last time in our notes we talked about, and we finished up our notes by talking about what were macromolecules. And that macromolecules are also known as polymers, two words that you should see on your vocab sheets. And they're made up of these smaller units known as monomers or subunits. And you also saw the word monomer in your vocab sheet as well. Now your body's going to need monomers to build larger molecules, the polymers, and your body's going to use these molecules to perform different functions that help you survive, that help you live. Now your body is going to get all of these macromolecules, these large polymers, from different food sources. And there's actually four main macromolecules or four main polymers that's going to be essential to all living things. Remember that when you have a molecule's first going to start off as, or the smallest part of a molecule will be an atom. The atoms will bond together, and those bonds will form those larger molecules. So that's what we're looking at today. We're actually going to look at the first two types of macromolecules that all living organisms need. And there's going to be four in total. So the first macromolecule that all living things need are carbohydrates. And a lot of times you'll hear people talking about eating or not eating lots of carbs. Carbohydrates are actually going to be molecules made up of the atoms C, H, and O, carbon, hydrogen, and oxygen. When you see C, H, and O, hopefully you also think of the word organic. Last time we talked about how any molecule that's made up of C, H, and O, CHO, is going to be known as organic. Those three elements are going to be bonded together. They're going to form a specific shape or polygon. And the monomer that makes up the carbohydrate is called a monosaccharide. Monosaccharide. So what does that mean? That means you have these three elements. They're going to be bonded together. And once they're bonded together, they're going to form this monomer, this subunit, and the special name for it, or the specific name for it, is monosaccharide. And a bunch of these monomers, or a bunch of these monosaccharides, are going to make a carbohydrate. So some examples of small carbs are glucose and sucrose. So these are just two more names of different types of monosaccharides. These are actually going to be used for quick energy sources for your cells. Before we continue, let's make a few little notes here. C, H, and L, this means that it's organic. Okay, all four of these macromolecules that all living things need are going to be organic. They're all going to contain C, H, and O. The monomer is called monosaccharide, so if we have a chain of them, a bunch of them will make the carbohydrate. Here we have your quick energy source. It's really important for you to know this as the function of carbohydrates, so please make this note on the side as well. Being a quick energy source is a function of carbohydrates, or what we use carbohydrates for. An example of large carbs are going to be starch and glycogen, so two more names of big carbohydrates. Glucose and sucrose are going to be small, and then starch and glycogen are going to be large. Starch and glycogen are going to be longer energy because they are actually bigger. Here you see one, in your notes you actually have the same pictures right here. Here you have glucose and sucrose. These are monomers, and you'll want to label that. This is a monomer, and notice that here you have monosaccharide, meaning it's made of one sugar or one monomer of the carbohydrate. Here you have the word disaccharide, and that's two sugars connected together. And sucrose is actually going to be the type of sugar that's sweet. Over here you have this underneath these two pictures in your notes, and this is a polysaccharide. Remember that poly means many, di, we just said, means two, and mono means one. Many sugars are going to make up a molecule like starch, and starch, remember, is for longer energy. So there's more little monomers, more little subunits that can be broken down to use for energy. So anytime you eat a carbohydrate like bread, pasta, anytime you eat a carbohydrate, your body is actually getting this larger molecule, this polysaccharide, and as you're digesting the food, your body's going to break it down into disaccharides or monosaccharides, eventually to the smallest subunit, and that's when you can actually use it for energy, for quick energy. Now, plants have a special type of carb known as cellulose, and this big carb provides support for plants as part of their cell wall. So this is the actually second function of carbs as part of support or actually structure. One thing that we'll need to know for each of our, or a couple things that we'll need to know for each of our macromolecules, our large molecules, is what atoms are they made up of? What is the name of their monomer? In this case, it's monosaccharide. What are their different functions for quick energy and for support or structure? And the last thing that we'll need to know for each of them is what is an indicator test for them? What is a test that we can use in class that can actually tell us if this molecule is present or not? For example, if you had a mystery mixture, or if you had some sort of solution or some sort of food, and you wanted to figure out, are there carbs in the food? For carbohydrates, the test is going to be known as Benedict's. And Benedict's is actually going to be able to indicate or detect that glucose is present. For carbohydrates, there's two tests. The first is Benedict's, and the second is iodine. That's number 2. For Benedict's, the chemical is actually a blue liquid, and if it detects glucose, you actually have to boil it for a little bit, and we'll actually get practice with that in class later on. If it actually has glucose in it, it'll turn from blue to orange. The second type of test is iodine, and this is going to indicate if starch is present. Okay? What was the difference between glucose and starch? Well, glucose was small, it was a monomer, and starch was a polymer, okay, a polysaccharide. It was a larger. And it's actually going to turn from a yellow-orange color to blue-black if starch is present. So these two tests are going to tell you whether or not the carbohydrate's there, if the glucose, the small carbohydrate, is there, or if starch, the large carbohydrate, is there. They're both going to start off as one color. They're both liquids, blue, or the yellow-orange. For glucose, if glucose is present, we're going to turn orange. If starch is present, we're going to turn to a blue-black color. And so you'll also have to memorize that color change for the indicators. Okay. And the last thing that we're going to talk about today are lipids. Lipids are our second macromolecule. It is also going to be organic. It's made up of atoms of C, H, and O. And the monomer or subunit for a lipid is known as a triglyceride. Okay. Here in your notes is actually, actually not yet. So lipids are going to have long tails called fatty acids. In your picture right here, you have a picture of that molecule and a picture of those elements that make it up. If you look really closely, you can see that for this lipid, you can actually see CHO, CHO, CHO. So there are the elements that are making it up. At the bottom half here, we have the fatty acid tail. And at the top here, we have what's going to be known as your glycerol. And for this specific structure, this is actually going to be your phospholipid. And we'll get to that in 1 second. But for lipids, lipids are also known as fats. Lipids can actually be differentiated in 2 ways, as saturated or unsaturated. If you're an unsaturated fatty acid, that's going to mean that you have kinks in your tails. So if you look at this picture here, you have one tail that's straight and one tail that has a little bend or a kink in it. And I'll label that as the kink. Any type of fat that's unsaturated or has a kink in it can actually be liquid at room temperature. So think about what type of fats we know of that's liquid. Well, most of that's going to be your oils, like your plant oils, vegetable oil, olive oil. All of those are liquid at room temperature, and that's because their molecular structure, their molecules and the bonds that they have actually form a little kink. And so that molecule is free to move around. Now, lipids are going to be important sources of long-term energy. So this is the function, or the important function that we need to know for lipids. Long-term energy, different from our carbohydrates. Carbohydrates were quick sources of energy. And they are often stored by animals and can serve as insulation. So our 2 functions, one is long-term energy. You do have to know that it's long-term and not quick. And number 2, it's going to be used as insulation. So if you think of a polar bear, okay, having a lot of fat in it. So last thing that we have is that the most important type of lipid, or the most important type of fat that we need to know of, is going to be your phospholipid. And that's what this is a picture of. So please make sure you label this as a phospholipid. Your phospholipids have a very unique shape, and that shape actually allows them to form the cell membrane around the outside of every cell. So right here you have your phospholipid. A bunch of these together are going to form our cell membrane because it's also a good example of our unsaturated, our straight fatty acid tail, and our unsaturated, our bent, our kink in our fatty acid tail as well. Here you actually see a picture of a cell membrane. And here at the top, you'll see those little circles that represent the phospholipid head. And underneath, you'll see the other head. So we'll actually talk more about this structure later on. But the last thing that we do need to know about lipids is that the indicator test for lipids is known as the brown paper bag. So if you think about eating at a fast food restaurant, if there is ever a brown bag, if you think of like a restaurant like Five Guys, you can, after you have your fries and your burger sitting there for a while, if you pick up the bag, you can actually see like a grease spot. Now that grease spot, if you actually took a small piece of it and held it up to the light, it would actually become a little bit transparent, a little bit clear. So here is our final test, or our only test, for lipids, the brown paper bag test. The other thing you want to think about here is that the unsaturated fats are going to be liquid at room temperature, but our saturated fats or lipids are going to be solid at room temperature. Okay, and we're going to go ahead and stop here for today. And just make sure you take a minute to look over your carbs and your lipids and answer your CYK.